بسم الله الرحمن الرحيم. Assalamu alaikum and good morning, my dear students of grade 11. I miss to hear from you all, and I hope everything is gonna be alright. And you are considering the previous sessions as it's really important for you all. To come directly to the session, we discussed last session the DNA replication, and before that we checked about the model of the DNA of Watson and Crick. And I just discussed with Mr. Shawqi, and he told me that he has a model. I checked it, exactly it's this one. You see you see it's bihelix, as you see, and you see it is coiled. You understand? You see it's how it's looking like. And this is the nitrogenic base of cytosine, guanine, adenine, thymine, cytosine, guanine, adenine, thymine, guanine, cytosine, thymine, adenine. It is like this. Okay? You see it now? I think it is looking clear right now. Okay? Yes. Like this. Okay. Today, we have the third session that is discussing DNA, RNA, and protein. And DNA codes, or what should be called codons, it is the codons that is making the RNA and RNA which guides the protein synthesis. Okay, so we're going slowly, step by step, to read and understand what's going on around us in this session. Look here, please. Like usually, essential questions. How are messenger RNA, ribosomal RNA, and transfer RNA involved in the transcription and translation of genes? This is meaning that we have two stations, two processes, two steps before making the protein, which called transcription and translation. What does mean transcription? What does mean translation? We are going on to study it together. It is something amazing. Just you need to concentrate 1000%. By the way, there is no 1000, there is no 100.1%. It's 100%. But just I'm showing you that it's really important. Okay, second question. What is the role of RNA polymerase in the synthesis of messenger RNA? There is RNA polymerase. This is what? This is enzyme, exactly. In the synthesis of messenger RNA. Okay, third question. How is the code of DNA translated into messenger RNA and utilized to synthesize a protein? I mean, how can the DNA make a protein? From DNA, how can we create a protein? Is there any stations? Is there any processes? Is there any factors that are making this? Exactly, yes. Remember this. We are going on to explain it. That there is a nucleus that contains DNA, and this is, as you know, it is a nucleus. And then there is a cell, the rest of the cell, and here there is ribosomes. This is ribosome. And there is a trip. And there are something happened between DNA and, and the ribosomes. What are the ribosomes? This is the factor of transcribing and translating the message that is coming from DNA. Message. So the message is coming with a messenger. Yes or not? This messenger is called messenger RNA. Step by step, we will know everything. So inside the DNA, inside the nucleus, there is DNA. This is the eukaryotes. The DNA is sending a signal to the ribosome. This signal is coming by the messenger RNA. So the messenger RNA is coming here. By the way, the ribosome is two bodies, the upper one and the lower one. And between both of them, there is a reader of the codons. What's reader of codons? We will know everything step by step. The reader of codons. This reader is called tRNA, transfer RNA. The transfer RNA is waiting here like this. Okay? Then the DNA, the messenger RNA is coming to be reading. So it is coming from exactly the nucleus to enter here from here. So it is moving. It is, for example, we'll discuss this step by step. T, C, G, T, C, G, A. So it is going to pass through this, exactly, this is codons, okay? It should be, has receptors on the T or in the A. That is reading this, so this is T, thymine, so there should be here what? Adenine. Waiting. Then the next, adenine. Then the next, guanine. Then the next, cytosine. Then the next, thymine, like this. So there is a reader for the codons. There is a reader which called tRNA, and there is a message coming from DNA by what? By mRNA, messenger RNA. By the way, mRNA is called messenger RNA is called mRNA. Ribosomal RNA is called rRNA. Transfer RNA is called tRNA. All this we'll discuss it step by step altogether today, and if this session will be not enough, we'll move to the next session, inshallah. So now we have 3 questions. First, I repeat it. How are mRNA, rRNA, tRNA involved in the transcription and translation of genes? That's just, I give you a brief, I just try to draw it to show you briefly the mechanism, but we'll discuss it step by step altogether. What is the role of RNA polymerase, this enzyme, in synthesis of mRNA? How is the code of DNA translated into messenger or mRNA and utilized to synthesize a protein? That's great. Okay. We have some vocabs, as you know. Synthesis, I think we are, mashallah, we are almost adult now. Synthesis here means to create something. To create something. Okay, we are calling photosynthesis. Means create food by the light. Photosynthesis. That is happening in the leaf of the plant. Okay? Synthesis means create something. So here we are discussing the creation of protein inside the cell. How protein is created. Look at your salivary glands. The cells are producing this saliva, amylase, in your mouth. Okay? This is enzyme that help for digesting partially carbs and something like this. Okay? Also, it is making the mouth full. There is easy or facilitation for chewing the food and facilitation for the tongue to move, and everything is becoming clear in this region. Okay? So the cells here are producing this enzyme. This enzyme means what? Means protein. Enzyme means protein. Okay? So this protein, how it is created, how it is synthesized inside these cells to be secreted in the mouth. all this we will discuss it today. So synthesize means create something. So what is RNA? What is mRNA? What is RNA? What is tRNA? What does mean transcription? What is this enzyme? RNA polymerase. What is intron? What is exon? What is codon? What is translation? Very, very, very important. All this, you need it, I swear, you need it all in your future when you will be in the medical college. All this, you will study it more deep in biochemistry, my family. Okay? That's great. Let's come here to discuss the central dogma. What is central dogma? Let's come to read it slowly to understand it. After the discovery of DNA structure by who? Who discovered it? Watson and Crick. Okay? The model of Watson and Crick. The scientists turned to investigating how DNA served as a genetic code for synthesis or the synthesis of protein. How DNA can make protein? It is a serious question. We are always saying, yeah, the body is well, blah blah blah, and there is a protein. Okay, how? The mechanism, how this could happen? You have to understand. Okay. Geneticists accept that the basic mechanism for reading and expressing genes is from DNA to RNA to protein. As I told you, it is a steps. Okay? It is a steps. Still one more time. These are the knuckles. This is DNA. So from DNA to RNA by on its way to mRNA. So it is a kind of RNA. To RNA ribosome. To tRNA, transfer RNA. All of them are RNA. Then there is what? This is called what? Ribosome. This is the factory of making protein. So from DNA to RNA, then finally there is a protein. You got the idea now? There is what? A protein. Okay. Just by the way, to think well about this. This is referred to as the central dogma of biology. DNA codes for RNA, which guides the synthesis of protein. So, what is the central dogma of biology? It is the DNA codes for RNA, which guides the synthesis of a protein or for the proteins. I hope it is clear now. So now we are going on to discuss the whole dogma by all what's happening inside this film, this whale. We want to know what's going on inside. All of it. Okay. We are starting by RNA. Attention, please. RNA is a nucleic acid. What is the difference between DNA and RNA? If you remember, we said DNA means deoxyribonucleic acid. Means the ribosic nucleic acid that is lack of oxygen. D means lack. Deoxy, lack of oxygen. Okay? But RNA, the ribosic. Okay? Ribosic nucleic acid. So RNA is nucleic acid similar to DNA, but with sugar ribose and with uracil instead of thymine. So there is a uracil instead of thymine. Okay? So if DNA, look here please, if DNA, the sequence of one of the genes is looking like this, thymine, adenine, thymine, C to Z, guanine, so exactly it should be here what? Adenine, thymine, adenine, guanine, C to Z. It should be like this, yeah? And this is in DNA. DNA. Let's make a copy of this, but RNA should be what? Thymine, I replace it with uracil. So it should be, look here, uracil, adenine, adenine, uracil. Okay, uracil, adenine, C to Z, guanine, guanine, C to Z. Okay, so this is DNA, this is RNA. So if I give you two models like this and ask you which one is DNA and which one is RNA, it is clear. When you see uracil with closed eye, this will be RNA. Okay, so be careful. I can give you several models of this and ask you what is correct and what is incorrect sequences. So you have to check the sequence. This is DNA or RNA first. Check. Thymine, adenine, adenine, thymine, thymine, adenine, cytosine, guanine, guanine, cytosine. That's great. Okay? So this is correct. And this should be DNA. But if it is uracil, adenine, adenine, uracil, uracil, adenine, cytosine, guanine, guanine, cytosine, this is a correct sequence also. But this is what? This is RNA. Attention, please. This is really serious. Okay. There are three types, you have to know it, of the RNA. There is mRNA, messenger RNA. What is messenger RNA? From its name, messenger. Messenger means coming with message. From where and to where? From where? From the nucleus. Why from the nucleus? Because there is DNA inside the nucleus. So the RNA is copied from the strand of DNA with a message to go outside the nucleus through the nuclear membrane to go to the cytoplasm to have a little migration or a transfer this message to what? To the ribosome, the factory of making protein. This is, please concentrate well. So let's come to read. mRNA look strands of RNA that are formed complementary to one strand of DNA. You see? You see? Direct synthesis of a specific protein. So it is going to make saliva. It's going to make amylase. It's going to make pepsinogen. It's going to make trypsinogen. Okay? All that enzymes are, as you know, are kinds of proteins. Okay? Kinds of proteins. Okay. Ribosomal RNA or RNA. Attention please to know what its function. It's very easy to give you the exam. Compare in a table between mRNA, rRNA, tRNA. Attention please. rRNA associates with proteins to form ribosomes in the cytoplasm. So what is forming ribosomes? RNA. Okay? It is looking clear now. So who is building the factory of making protein? It is RNA. That is associates with proteins to form ribosomes in the cytoplasm. I think it is looking clear. Now with the third one, tRNA. Small. It is a smaller segment of RNA that transport amino acids to the ribosomes. Transport amino acid to the ribosome. What this amino acid doing, as I show you It is reading the sequences, okay, and making a copy of it, which is called exon and introns. We will discuss this step by step. Cool. Transcription. We want to know what this means, transcription. Transcription. First step of the central dogma involves the synthesis of messenger RNA. So the first step is to create RNA, the messenger one, from one strand of DNA. From DNA in a process called transcription. So what is transcription? It is a process happening in the nucleus that is synthesize the messenger RNA from one strand of DNA. Clear now? Look here. The enzyme RNA polymerase. Now we came to focus the mechanism itself. Now we just identified the mechanism. Let's come to see the mechanism itself. The enzyme RNA polymerase regulates RNA synthesis by binding to the specific section where a messenger RNA will be synthesized. Look, this is the RNA polymerase. Okay, it came to a specific place that is ordered from Allah subhanahu wa ta'ala that to be in this region. Okay? RNA polymerase moves along DNA strand in 3 to 5 direction, from terminal 3 to terminal 5. Consider it well. Last time also we discussed this, that it started from terminal 3 to terminal 5. Cannot start from terminal 5 to terminal 3. Okay? Look what's going on. It is moving here. Okay? On one strand, not on both. In one strand only. Okay? Okay. RNA polymerase moves along DNA strand in a 3 to 5 terminal direction, synthesizing messenger RNA. So it is coming with, so you can just think about it or look at it from your way, that it is just an empty strand with this RNA polymerase and just passing through one strand and taking a copy. So cytosine will be guanine, guanine will be cytosine, uracil will be unlike this. Okay? That's great. RNA processing. And comparing DNA code with its transcribed messenger RNA code, scientists found that the messenger RNA code was much shorter. Yes, so the DNA can be, this is one strand, okay? It's just one strand. One strand of the DNA. Okay? As you know, for example, here cytosine, guanine, guanine, cytosine, adenine, thymine, thymine, adenine, thymine, adenine, cytosine, guanine, guanine, cytosine, guanine, cytosine, and like this. So we see that the polymerase, RNA polymerase, is coming with an empty strand, but short enough. When it is coming with short enough, it is cytosine, so it should be here guanine. Guanine should be here cytosine. Adenine should be here what? Thymine? No, uracil. Okay? Should be what? Uracil. And like this. So, but it is short. It is not during the whole strand. No, it is short. Because it is coming with a specific function. To make a specific protein. Not to make a copy of DNA. If there is a copy of DNA, this is called DNA replication. This is something we studied it also before. But in short ways, okay, this meaning this is exactly synthesizing synthesis of mRNA. Okay, you got the idea now? I hope so. DNA sequences not found in mRNA are called introns. DNA sequences that remain in final messenger RNA are called exons. Attention please. The DNA sequences that not found in messenger RNA are called introns. But DNA sequences that remain in the final messenger RNA are called what? Exons. You got the idea? Okay. Scientists hypothesized that the instructions from protein synthesis were encoded in DNA. They were encoded in DNA. Experiments during the 1960s demonstrated that the DNA code was the 3-base code. The 3-base codes in DNA or messenger RNA are called codons. Are called what? Codons. Okay? Okay. It's looking clear. Yeah, it is really looking clear. Yeah. Okay. Now we are going to the translation. We are going to what? To the translation. We finished with the transcription, and we know that the transcription is the creation or the creating or synthesizing messenger RNA that is coming with the message. Then it is migrating from the DNA through the nuclear membrane to the cytoplasm. Then it's taking the direction of what? Of this. What is this? This is the ribosome. This is what? The ribosome. This is the ribosome. Okay. What is going on here? Attention please. After synthesis, messenger RNA moves from the nucleus into the cytoplasm. Okay. Where it connects at the 5 into the ribosomes. Connect through here the first time. Please concentrate well. It is the first time to discuss that it is connected from 5 terminal. Okay? The messenger RNA code is read and translated into a protein through a process called translation. Look at the translation. Look at this green strand. This green strand, this is the messenger RNA. No. This one is the tRNA. When it is passing, okay, this is the messenger RNA. When it is passing, coming from the 5th terminal, as you see, okay, passing from between the upper part and the lower part, or the upper and lower body of this ribosome, it is read exactly. So look here, adenine, uracil, adenine, guanine, cytosine. Uracil, adenine. Adenine should be what? Adenine should be what? Uracil should be here. Cytosine, guanine. Cytosine. You see? UGC. This one is called what? Anticodon. So it's coming by 3. 3 by 3. This is called what? Anticodon. that is coming. Each three of them, they are coming to read. What is here? It is uracil, uracil, uracil. So there is anticodon with adenine, adenine, adenine. Here is adenine, cytosine, guanine. This is called what? Codon. Each three of them are called codon. So this codon and there is the anticodon. Okay? The anticodon is created the protein through reading and translating this nitrogenic base, what's mentioned here. So, for example, I'll give you an example from myself. CGC. This is the codon. What is the anticodon should be? Huh? GCG. Yes or not? Yes or not? That's great. I hope you understand the mechanism now. This is the mechanism. Later you will study everything here when you will be in the college, inshallah. Okay. Give it a chance to see how many minutes we spent. Oh, 31 minutes. Okay, this will be enough for you today, and we'll continue inshallah next session. Good luck and enjoy your moment. Bye.